Hi, I'm Pablo Fonseca, and today I will present the results of the study entitled Meta-analysis of differentially co-expressed networks composed by genes and long non-coding RNAs associated with fertility status in Holstein cows. This study was developed at the University of Guelph with the advisory of Professor Angela Canovas. Subfertility is among the major causes of economic losses and welfare concerns in livestock species. To improve the profitability and animal welfare in the dairy industry, it's important to better understand the genetic architecture associated with fertility status. Integration of multi-level biological information such as genomics and transcriptomics has the potential to pinpoint key biological process, candidate genes, and genetic variants associated with poor fertility. Therefore, the objective of this study was to identify potential candidate genes and single nucleotide polymorphisms (SNPs) associated with the fertility status in Holstein cows through a meta-analysis and the integration of transcriptomic and genomic information. As material methods, we obtained two RNA-seq data from the Gene Expression Omnibus database from previous published studies. In these studies, animals were classified as high fertile and subfertile classes based on the distribution of the estimated breeding values for calf interval. So, briefly, the cows which were in the top 20% and in the bottom 5% for the distribution of the EBVs for calf interval were classified as high fertile and subfertile animals respectively. And at the end, we obtained 8 high fertile animals and 9 subfertile animals, which endometrium biopsies were performed and endometrial samples were submitted to RNA sequencing. And using a bioinformatic pipeline composed by transcript counts, likelihood ratio tests in order to identify transcripts which the expression were guided by the difference between studies and not between conditions were excluded, followed by detection of long non-coding RNAs and the detection of co-expressed gene network, which was performed using the waved gene co-expression network analysis, WGCNA, and also network meta-analysis to identify differentially expressed genes in both studies, resulted in the possibility to identify key regulators of the fertility status in this case. And these key regulators were genes which were top hub genes that explain the majority of the topology of these co-expression networks that were exclusively co-expressed in high fertile or subfertile animals, and also harbor genetic variants which were fixed in high fertile or subfertile animals. These variants mapped in these top hub genes were used in the VAP software in order to predict the functional impact in these variants over the function of the gene. And as results, we had the opportunity to identify 3 functional candidate models of co-expressed genes that were exclusively co-expressed in one of the groups that were upregulated in subfertile animals, and 4 models of co-expressed gene networks that were upregulated in high fertile animals. Additionally, the prospection for SNPs on those genes allowed the identification of SNPs that were fixed in high fertile or subfertile animals. In figure 1 and 2, we have an example of a long non-coding RNA gene co-expression network and the Z-score of the expression values for each one of the co-expression models identified in this study. The functional analysis of the candidate genes mapped in each one of the differentially co-expressed models indicated that those genes are associated to functionally relevant processes for fertility efficiency, such as regulation of hormone production, development of embryonic structure and tissues, implantation and embryonic proliferation, among other processes. As conclusions, the results obtained in the current study open new possibilities for future investigation of the regulatory potential of co-expression networks composed by gene and lognal coding RNAs affecting the fertility status in dairy cows. Moreover, identification of potential biomarkers such as long coding RNAs and functional variants in candidate genes may help to reduce the frequency of subfertility in the population, reducing the economic losses and increasing the animal welfare. Thank you very much, and I will be more than happy to answer any question. Thank you very much.